you very much to you as president of Inspire in Qatar for inviting me to chair this session. And it gives me great pleasure actually to introduce Professor Nick Embleton today. He's a consultant neonatologist and a professor in neonatal medicine in Newcastle in the UK. He's an extremely well recognized for his repertoire in neonatal nutrition, and he's also leading several research trials and projects at the moment and has extensively published. He's editor of pediatric research and is the UK representative on ESPGHAN, to name just a few of his many accolades. And I'm very, very pleased that he is sharing his time, knowledge and expertise with us this evening. And I think that he's going to talk today about the nutritional interventions to improve brain outcomes in preterm infants. So over to you, Nick.
Great. Thanks very much, Sybil. Thanks, Kieran. Thanks, Manish, for inviting me. Sybil says I'm going to spend the next 40 minutes or so talking about nutritional interventions to improve brain outcomes in preterm infants. It's a relatively small group of you, and I'm going to take some questions at the end, but you're welcome to email me. My email address is there if there are specific things that you want to pick up on, and we've got a research website, neonatalresearch.net, with some information on our various studies that we get involved in. And as Sybil says, we seem to have got involved in an awful lot of research in the last 10 or 15 years. We've been involved in lots of very large collaborative trials. Many of you will know the Sift trials and the Embleton trials. So that was about 5,000 babies we recruited. We've got a major interest in the gut microbiome, but also in the metabolome. So we're exploring new techniques with that. We've got teams looking at breast milk composition, so particularly IgA compositions, human milk oligosaccharides in breast milk, and also a team at Northumbria looking at bacteriophage viruses in breast milk. So a lot of our work is focused on NEC and the immunology and responses. Chris Stewart, who you can see there in the bottom left-hand corner, works in the lab and has set up a system of enteroids. So these are gut organoids grown from stem cells that we retrieve if the babies need surgery. We've got cohorts with long-term metabolic outcome, and Janet Berrington, who you can also see there on the left, coordinates our neonatal biobank, and in that we've got samples of stool, urine, blood and breast milk from over 1,000 preterm babies. So as I say, we're a pretty active research group with lots of studies going on. This is Newcastle, beautiful river, pretty town, and lots of lights and activity. We've got some beautiful coastline and castles, and we've got an amazing football team who never win anything at all, but they're still great fun to follow. So what am I going to be talking about? I'm going to be talking about actually what do we mean by nutrition, then talk a bit about brain growth and the relevance of nutrition, different types of interventions that may impact on brain development, and then finishing up on nutritional assessment. On the right here, you'll see some of my disclosures. Most of our funding has come from governmental sources or the research councils, but we have been funded by some commercial organizations as well. And if I get through all of that, then we can start thinking about some of the complexity of breast milk, which I just outlined there, and I'm going to come back to that right at the very end. So as I say, I think the starting point is to encourage everyone to realize that nutrition is more than nutrients. I mean, clearly the actual quantity of nutrients is critical for preterm babies, and that's true for proteins and fats, but also all your micronutrients. But I see another component here, which are all the functional aspects that are particularly associated with breast milk. So these may be human milk oligosaccharides or growth factors, although it could be enzymes in breast milk. There are now all these microbial aspects that we've really started only to appreciate, I think, in the last 10 or 15 years. And these are the microbes that are acquired from breast milk or promoted from breast milk components, but also the NICU environment and the role of probiotics. And then the last component of nutrition are all the, what I'm kind of group together as the kind of technical aspects, the socio-behavioral aspects. These might be things like, whether you're going to feed boluses or give skin to skin, how you breastfeed, what mothers think and do, sensory aspects. So I think those 4 components need to be seen as nutrition. So when we're talking about how nutrition improves outcome in preterm babies, let's use that as a schema. Now, when I started doing my research many years ago, I sort of imagined that human babies would grow pretty much like primates. In fact, we're very different to primates. So here, what I show you in this graph is that the rapid period of brain growth in most non-human primates occurs prior to term. In comparison, the rapid period of brain growth in preterm in human babies occurs in the 3rd trimester and the first few months after delivery. So that between 24 weeks gestation and 2 years of age, we acquire about 90% of our final adult brain volume. So the vulnerability of the brain to undernutrition comes at a very different period to most other animals. Here's another way of looking at it. Here's a rhinoceros with a huge head. And I could play a little game here and ask you to work out where the brain is in the skull of the rhinoceros, but I'll save time. And here it is. The brain of this massive animal is really quite small. Whereas in comparison, the human brain fills the entire cranial cavity. So we really have a very large brain. Another way of thinking about it, along the x-axis here, I show you body weight, and up the y-axis, I show you brain weight. This is a logarithmic scale. And you can see that most animals plot out somewhere on that average line. Whereas human babies are here, they're off the scale. And in comparison to most other mammals, the human brain is about 10 or 20 times as big. And everything about placental physiology, nutrient transfer, breast milk composition, everything is designed to protect and grow this brain. That's what we evolved to do. The reason we're in control of the planet is of our huge brains. And so all of nutrition is directed to that in early life. And none of this is about preventing us getting diabetes or obesity or something like that. Now, of course, although the brain acquires a lot of this volume in the 3rd trimester and the first few months, there are lots of different processes. So particularly active during all the 3 trimesters is neuronal migration. Apoptosis, particularly active in the 2nd and 3rd trimesters, but continues in the first few months after birth. Synaptogenesis that really only kind of kicks off in the 3rd trimester, but may continue over the first 1 or 2 years of life. And myelination, which may not be really complete until we're about 20 years of age. So there are lots of different processes going on, and we've started to understand that many of these are actually under control of growth factors, and particularly growth factors like IGF-1. And there is now increasing evidence that nutrients modulate the levels of IGF-1. And so there is now a direct link between how feeding our babies may impact on a range of brain developmental processes. Now, briefly, just to understand the biology, why is it the preterm brain is so vulnerable to poor nutrition? Well, all of us recognize, unfortunately, that damage is quite common in premature babies, particularly cystic PVL, but also hemorrhage. And repair of this damaged tissue is likely to require higher intakes. It makes sense that if you're going to repair the damage as well as keep on growing, you're going to need more nutrients. And so not only do you need more substrate, but you need more energy to power the process. As I say, we've got a very large brain and very high demands, and therefore even marginal malnutrition is quite easy to do. And then finally, we've started to realize that what we've been looking at may be missing some key aspects. So with an ultrasound scan, we clearly don't get a good look at the cerebellum. And even on a normal MRI, you don't really explore the cerebellum or the brainstem in great detail. But we now know that cerebellar growth is actually much more rapid than cortical growth in the last trimester. We can see differences in preterm babies compared to term babies. Interestingly, they have different shaped cerebellar and brain stems. And various research groups have documented the abnormal development that is occurring even when the MRI appears grossly normal. And some beautiful pictures from a study of Weir et al. Last year came out showing you the complexity and the changing shapes of the cerebellum. And many of you will also be aware of the devastating longer-term consequences of diseases such as NEC and sepsis, this cytokine storm that is initiated. And again, we're starting now to understand how this is provoked. And generally, a lot of this involves the toll-like receptors that sit in lots of tissues, but particularly sit on the surface of the microglial cells. And TLR activation results in production of free radicals and cytokines. And somehow these cytokines seem to damage these oligodendrocyte progenitor cells. So babies are then going through a process of disturbed maturation. There's also damage to axons or neurons. And furthermore, these cytokines seem through, again, a whole series of mechanisms to suppress growth factors, and particularly IGF-1. So how might nutrient deficiency impact on the brain? Well, we could think about three hypothetical nutrients here, where the severity or the timing might differ. So, for example, a deficiency of nutrient A might particularly impact on these neuronal processes, neuronal migration, whereas nutrient C might have a larger impact on synaptogenesis and myelination. And therefore, the difference between A and C is going to result in a different phenotype. So not all nutrient deficiencies are going to cause the same problem. An example of a nutrient deficiency like type A might be, for example, macronutrients. We know how difficult it is to achieve and maintain both parenteral and enteral nutrient tolerance in the first few days, and many of our babies don't get what they need. So macronutrients may affect these processes. An example of a nutrient that may not be apparent right from the beginning, but may appear at a few weeks of age, would be something like iron. Quite often we forget to start iron at the important point in time. Whereas nutrient C might be something like DHA or choline. We know that IV lipid solutions are so optimal. We know that maternal diets actually now don't contain very much DHA. In breast milk, DHA concentrations are quite low. And therefore, nutrient C might have an impact that is less severe, but over a much more prolonged period of time. So you can see that each of these three different nutrients are occurring at different points in time, different severities, there are different developmental stages, and there are likely to be multiple mechanisms. So the end result phenotype is going to differ quite a lot. And again, remember that none of these nutrients work in isolation. They all require cofactors, enzymes, and energy. And a healthy brain needs every single nutrient in the diet. It's not just that you need zinc or DHA, of course, you need every other nutrient as well. So just to kind of put in context the enormity of macronutrient requirements in preterm babies, this is Egan Bernal, a Colombian cyclist who won the 2020 Tour de France. And he cycles up and down mountains for a living for 3 weeks on end. And whilst he's doing this, he's consuming around about 7,000 kilocalories a day. Just imagine how wonderful it must be to consume that many calories and just not get fat. So he's expending around about 100 kilocalories a kilo a day, and that allows him to cycle up and down mountains for hours at an end. But in comparison to a baby on the neonatal unit, where we know that energy requirements are somewhere in the order of 120 to 130 kilocalories a kilo a day. So we are looking after babies who have an energy requirement that is about 20 or 30% greater than that is required to cycle up and down mountains. And remember that more than half of that energy expenditure is in the brain. And when you start to appreciate how large these requirements are, I think it's easy to understand perhaps why brain development might be suboptimal in our babies. So lots of different ways that nutrients might be important for brain development. Of course, we need nutrients for tissue substrates, all of your macronutrients and micronutrients. We need energy to drive the system, so that's particularly carbohydrate and lipids, but also you'll burn your protein if you don't give enough other energy sources. There are all these signaling and growth factor impacts of nutrition, so particularly everything regulated through mTOR, but also all the lipids associated with the milk fat globule membrane, IGF-1, EGF. Lots of nutrients seem to have impacts on gene expression and DNA methylation, so particularly folate, B12 and iron, and many others. We know that breast milk reduces the risk of diseases such as neck and sepsis, and both of these do result in worse brain outcomes. And then finally, there's the whole role of the gut microbiome and all the various prebiotics, HMOs, and other substances like lactoferrin. So these 6 different kind of groups of mechanisms that link nutrition to the brain. So this is a picture from the neonatal unit that those of you who've been lucky enough to work in Newcastle will recognize. This is the back shot, a picture I took a couple of years back, and just look at this complexity. And these are all the things that you think about and talk about every day on the ward round, you know, whether you should put the baby under phototherapy, should you put a long line in, how much fluid, which inotropes should you use, what's the right level of oxygen saturation, how long do you keep the antibiotics going. This is the stuff that occupies us day in, day out on the ward round. And this is an incredibly complex environment, but actually within all that complexity, the focus tends to be on cardiorespiratory care. There's relatively little focus on nutrition. Like none of those monitors are monitoring anything to do with nutrition. So, brief pause for breath, quick recap. Nutrition is more than nutrients. Brain growth is very rapid, but we only have one brain for life. The impact of nutrient deficiency on the brain depends on the amount, the type, the timing, and the duration of any deficit. The NICU is a very complex environment, and poor nutritional status is very common. And when we come to think about poor brain outcome, then should we be thinking more about macronutrients or micronutrients? Now, this is work that I did many years ago now, but it elaborates a kind of key principle. So these yellow bars represent a 3 gram per kilo protein intake that a baby should receive on each day of the first 14 days. Now, of course, we now tend to recommend, if you're feeding babies enterally, around about 4 grams a kilo, but we weren't quite as ambitious back 25 years ago. So those yellow bars represents the 3 grams, the recommended intake that a baby should have each day. And back then we were still starting feeds pretty early. We were getting to full feeds in these babies generally by about day 10. So we were doing a pretty good job even 20 years ago. And we felt pretty pleased with what we were achieving. When you actually stop to look at what babies are getting in protein, that is depicted in the green bar here. And what you can see is that most babies don't get their protein requirements on any one of the first 14 days of life. So the difference between the recommended in yellow and the actual achieved, the actual intake in green, is the red bar. The red bar represents on day 4 the deficit in nutrient that baby needs. And if you add that over time, this is what you get. You get this cumulative deficit of protein and energy with lots of other micronutrients that just keeps on growing without you realizing about it. You think you're doing a great job, but actually we're busy malnourishing the babies every day on the neonatal unit. So we need to do a much better job in terms of being aware of where we're getting. And this is the problem. We have these monitors here that give you second by second feedback on the cardiorespiratory system, but there are no monitors around that are telling you how good a job you're doing with your nutrition. And so malnutrition is largely invisible. We don't have any feedback mechanisms to remind you. We don't have any alarms that go off if you're not meeting a baby's protein requirement. So what's the evidence that macronutrient requirements matter? So I'm going to show you 5 different studies, but I could have shown you 50 different studies. The first one looks at first week macronutrient intakes. The next one is about energy intakes and ROP. The next study looks at MRI data at IQ at 16 years, and then some more detailed MRI. So these are just 5 studies, but I could have picked any one of several larger studies. The study from the States, more than 10 years old now, 124 babies weighing less than a kilo. They started PN early, but they were quite slow to increase, and they used multiple regressions to adjust the data for all the likely confounders. And the question they asked was, what was the association between protein and energy intakes in the first week of life, the first 7 days, and development 18 months later? And this is what they found, that for every extra 10 kilocalories a kilo a day energy intake that they achieved, there was about a 4.5 point improvement in mental developmental index on the baby at 18 months of age. And the same pattern, that for every extra gram per kilo of protein in the first week of life, about an 8-point advantage in developmental outcome at 18 months of age. So this data was all adjusted for sex, maternal educational status, NEC, and all these other factors. Even having done those adjustments, there's still a correlation between the amount of macronutrients and your brain development 18 months later. This is a large Swedish study from the Express cohort, almost 500 very premature babies, less than 17 weeks, less than 27 weeks. They again found that intakes were much less than recommended, but what they did see was that the higher intakes of fat, carbohydrates, and energy were associated with a decreased risk of retinopathy. So here I've graphed it out here, and what you can see is that for every 10 kilocalorie a kilo a day increase that the baby has over the first 4 weeks of life, so this is parenteral and enteral, there's about a 24% reduction in ROP after they control for all known confounders. And it's likely that low energy intake is acting through multiple different mechanisms. Partly it's suppressing IGF-1, but partly it's also arresting vascularization. So a complex series of mechanisms, but very clear evidence that energy intake relates to ROP. This is a Norwegian study, a relatively small study. Again, this was a parenteral and enteral study where they gave one group of babies much higher intakes of amino acids, lipids, and DHA, and then they conducted MRI at term. What they showed was that in the intervention group, weight gain was better, 16.5 versus 13.8 grams per kilo a day, and head growth was better. But most impressive were the findings in the white matter tracts on the MRI, and they looked particularly at this part of the brain called the superior longitudinal fasciculi, which is involved in motor function, perception, and language development. And this is what they saw. So here is the MRI, and in green picked out is the normal white matter skeleton. But then picked out in red and orange is the difference in the superior longitudinal fasciculus between the two groups of babies, the higher nutrient and the standard nutrient, after they adjust for birth weight and age. So clear MRI evidence of a difference in growth in just a few weeks of time on the neonatal unit. Many of you will know about the work of Alan Lucas. And here, this is a follow-up study looking at 76 children, either randomized getting a standard nutrient diet or a higher nutrient diet, so either term formula versus preterm formula. This cohort consists just of children who are neurologically normal at 8 years of age and who underwent an MRI and a Vessela later on. So here are the data at 16 years of age, and what we see is that 16 years later, after an intervention that only lasted for an average of 4 weeks, there's an 8-point higher verbal IQ in those on a higher nutrient diet. Overall, there were no differences at 16 years of age in total brain volume, but there were significant differences in the size of the caudate. And we know that the caudate is particularly vulnerable, therefore, to undernutrition and environmental factors. And here is some very recent evidence, came out last year. Again, an observational cohort of 178 babies around 26 weeks gestation, comparing it before and after. In the earlier cohort, just 2.7 grams on average of protein, whereas in the later cohort, 3.4, and a small difference in energy. And they looked at the MRI, and what they showed was that at 30 weeks gestation, there were significant differences in a whole range of brain regions. But by term, it was really the caudate again that appeared to be different between those on the higher nutrient intakes in the later epoch compared to the earlier epochs. So hopefully I've convinced you about the importance of macronutrients. Strong evidence that these relate to short and long-term brain outcomes. As I say, we need to accrete lean tissue and get enough energy to drive the system. But what about micronutrients? What about some of these factors here? Well, taurine and glutamine are key amino acids. Taurine, conditionally essential amino acid, present in very high quantities in the brain. There is some observational evidence that lower levels are associated with worse developmental outcomes, but there's really only been very small RCTs, and we're not really convinced that supplemental taurine is beneficial. Glutamine has also been looked at in a small trial, but no effect. So I think our conclusion at the minute is that the data for giving extra amino acids, specific amino acids, are fairly weak. No good evidence that giving additional amino acids as supplements are beneficial for the brain. But of course, there are good theoretical reasons why amino acids might be important, and a lot of these effects might be modulated through mTOR signaling. What about iron? Iron is probably one of the commonest nutrient deficiencies globally. It's essential not just for blood cells, but for myelination and neurotransmitters. And preterm babies, not surprisingly, at very high risk. They have relatively low iron stores at birth with very high growth velocity, very low levels of iron in breast milk. And of course, babies lose a lot of iron in all the blood tests. Very strong RCT data that anemia in childhood results in worse cognitive outcomes. But we do have to be careful. We don't actually possess a mechanism to excrete excess iron. And iron, of course, is very pro-inflammatory. So we do need to be careful. However, there are no really good RCTs in preterm babies, but very good theoretical reasons why we should be making sure that babies get sufficient iron from about 3 or 4 weeks of age. And again, you only need around about 2 or 3 milligrams a kilo a day. You might need more if you're giving babies EPO supplements. Our own practice is that you could probably delay iron supplements if the babies have been transfused in the last 7 days, or if they've got a particularly high ferritin. We don't tend to measure ferritin routinely, but if you're unsure, you can always measure it, and if it's very high, you probably don't need to start the iron for another week or so. What about some of the other metals and micronutrients? Well, not surprisingly, zinc is involved in just about every body system in the body. It's involved in particularly development of certain brain regions, hippocampus and cerebellum. There are lots of interactions, complexity between some of these metals, interactions between copper and iron. Iodine, another key nutrient, of course, needed for the brain, although unfortunately the largest RCT a few years ago didn't show any benefit of giving extra iodine. So for all of these key micronutrients, there are no good RCTs. It's likely that current intakes are probably going to be sufficient, with the exception of babies on prolonged parenteral nutrition. By that I mean babies on pure PN for more than 2 or 3 weeks. But I think routinely we don't need to give additional supplements of those metals. So we've talked a little bit about the importance of macronutrients on the micronutrients, the range of different components in breast milk and how these might be important. So all of these 4 last factors are particularly relevant to how human breast milk may promote good or better brain development in preterm babies. And that goes particularly for all of these complex phospholipids, lots of aspects of gene expression, but also all the microbial aspects and the prevention of disease. So how strong are the data? Well, this is again data from Alan Lucas's group looking at adolescents at 15 years of age who are neurologically normal. The largest predictors of IQ in these studies were social class, but also breast milk intake. And the amount of breast milk babies received on the neonatal unit was associated 15 years later with verbal and full-scale IQ, white matter volume on the MRI. And there appears to be a dose response effect. The more breast milk these babies get, the better those outcomes were. This is data from a large French cohort, almost 3,000 babies, less than 32 weeks. And what they noted in this study is that babies on breast milk seem to have slower weight gain. They talk about this breastfeeding paradox, whereby breastfed babies might grow more slowly but have better developmental outcome. So on the right here, up the y-axis, you've got a Kaufman ABC, a measure of neurodevelopment at 2 years of age, and then 4 cohorts. So 880 babies who didn't receive any breast milk on the left, and then 91 babies who were still being breastfed 2 months after discharge. And what you can see here is a fairly convincing linear relationship. So the longer you receive mother's own milk, both pre and post discharge, the better your brain outcome is going to be. So what about donor milk? If we haven't got enough mother's own milk, does donor milk improve brain outcomes? So this was a trial conducted by Deb O'Connor, published 3 or 4 years ago now, 4 NICUs in Canada, almost just over 350 babies, where they used donor milk or formula to make up any shortfall in mother's own milk supply. And these babies with a mean birth weight just under 1,000 grams. The intervention lasted for 90 days, and the primary outcome was neurodevelopment. And here is the data, outcome data. Here in the light blue is the donor milk, and the dark blue is formula. And what you can see is that neither for the cognitive, language, or motor domain is there any advantage, long-term advantage of using donor milk. If anything, the data seem to hint a little bit towards the fact that formula-fed babies might do a little bit better. And that may just be a chance finding, or it may be that babies on the formula were actually getting high levels of macronutrients. So I don't think at the minute there's any evidence to suggest that a reason for using donor milk is that you're going to improve brain outcomes per se. There may be other benefits of using donor milk in terms of avoiding NEC. Now I just want to move on to another kind of key group of components. These are the long chain polyunsaturated fatty acids, particularly docosahexaenoic acid, DHA, and arachidonic acid. And we know that placental uptake of these is particularly active in the third trimester, which babies miss out, preterm babies miss out on. So preterm babies don't get as much of the placenta. They have very low fat stores and therefore low stores of these key long-chain fatty acids. But they also have pretty immature digestive metabolic functions. They might not be absorbing these lipids optimally from the GI tract. And as I mentioned previously, we know that intravenous lipid composition is far from ideal. There is a Cochrane meta-analysis on this. Generally, the quality of the trials is pretty good, just over 2,000 infants in 17 trials, but overall they showed no benefit of supplemental DHA. However, these studies are pretty heterogeneous. They were all really underpowered to look at brain outcomes at 2 years of age. Lots of differences in assessment and methodology, source of DHA and composition, and mainly these were the healthier preterm babies who were formula fed. So I think at the minute there's no justification based on that meta-analysis to go dumping extra DHA into formula milk, but clearly these trials haven't really been conducted with the aim of looking at long-term brain outcome. So strong theoretical benefit of having DHA. Should we just give the babies extra DHA to high-risk babies? Why not just give them some? It's not that expensive. Why don't we just give them a bit extra to be on the safe side? Well, I think these studies suggest that we need to be cautious in terms of how we approach it. So this is from an Australian group, Carmel Collins's group, published in New England Journal 3 years ago, where they actually randomized babies at risk of BPD to either getting supplemental DHA or placebo. And what they hypothesized was that babies getting DHA would have a decreased incidence of BPD. In fact, they showed the opposite. So the suggestion here that the babies getting DHA actually had a higher rate of BPD. And that might be because babies were just getting DHA, they weren't getting additional arachidonic acid or choline or anything else like that. So a note of caution there, and that's been replicated in a study last year from a Canadian group published in JAMA, where they thought that the safe way of doing it might be to supplement the mother. So the mother was taking DHA supplements with the intention that we might see a decreased rate of BPD. In fact, again, there was no evidence of a reduction in BPD, and in fact the trial was stopped early because it appeared that outcomes might be actually slightly worse in the DHA group. So I think this suggests we need a lot more data before we start randomly putting DHA into babies' feeds. We need to carefully consider the ratio of N3 to N6 fatty acids. DHA is clearly essential, but simply adding more isn't going to be beneficial. However, there is cause for optimism that additional fatty acids may result in benefits. So this is a study from Ann Hellström's group in Sweden, who have a very long track record of research into retinopathy, IGF-1, and these type of studies. And this study published earlier this year in JAMA, 207 babies less than 27 weeks gestation randomized to getting additional arachidonic acid and DHA from day 3 until they reached the corrected age of term, compared to placebo group. And the outcome was severe retinopathy. And what they saw was that in the intervention group, the rate of severe retinopathy was about halved. So 15% in the intervention group compared to 33% in the control group. They were also able to show higher amounts of arachidonic and docosahexaenoic acid in the serum phospholipids. And in this study, they showed no difference on rates of BPD, IVH, or sepsis. And on the right there, you can see a sort of an upside down Kaplan-Meier, showing again quite convincingly separation between the control group and the intervention group. So I think there is cause for optimism. And I want to sort of start finishing off by telling you about a trial that we will be starting next year. And this involves giving a supplement that also contains something called choline. Now, choline is one of these nutrients that most people who aren't interested in neonatal nutrition haven't really kind of heard much about. It's a bit like a B vitamin, but it's involved in lots of different processes, particularly fetal placental growth, but it's involved in DNA methylation. It's a precursor of acetylcholine, and it's involved in neuronal proliferation, migration. It seems to be involved in brain plasticity, synapse formation, and it acts as a substrate for phosphatidylcholine and sphingomyelin. As I say, it's a very important source of methyl groups for epigenetic modifications and therefore may have long-lasting effects. And there's some basic data in rats showing that maternal choline supplementation improves memory and attention in rats. So the mechanisms of choline action are going to be very complex, and they're going to involve neuroanatomical substrate, but also chemical mechanisms and molecular. Where do you get choline from? Well, you particularly get it from fish and eggs and meat and these types of nutrients. And if you actually look at most adult diets, many people probably are marginally under eating choline. And there has been a trial from people that I'm now collaborating with, where they gave babies extra choline in a supplement that also contained additional docosahexaenoic acid, DHA, and a nucleotide UMP. So this was a relatively small RCT, but it was conducted in babies who were both preterm or term babies with brain injury. And again, the primary outcome was at 2 years of age. And this is the data. In red is the baby at 2 years of age, in black is the baby at 12 months of age. And what you can see at the top there in terms of the cognitive outcome, it is just about significant. It appears that giving supplements of choline, DHA, and UMP for the first 1 year of life does improve cognitive outcome. It doesn't seem to have any impact on motor outcome, and it may or may not be impacting on language, but there does seem there evidence that there may be benefit on cognition. All of you will know that a trial of just 45 babies is never going to be sufficient to change practice, and so we've been working for the last few years trying to develop a trial. I'm pleased to say it has now been funded by the UK NIHR. And the trial unit, again, is going to be the National Perinatal Epidemiology Unit in Oxford, who've conducted many of our other trials. So we're now funded to conduct a follow-on dolphin study, developmental outcomes of long-term feed fortification in neonates. This is a double-blind RCT. We're going to be recruiting over 1,000 babies from 30 NICUs across the UK. We're going to be recruiting preterm babies less than 28 weeks or term babies with HIE. And they'll be randomized to either getting a supplement of DHA, choline, and UMP or placebo. And the primary outcome will be developmental outcome at 24 months of age using a Parker R. But there's a whole range of secondary outcomes, strength and difficulties, questionnaire, age and stages, NHS data, health economics, and permissions to track these children's education up until the age of 11 years of age. And anybody who's listening and currently working in the UK who might be interested in being a recruiting unit, you're very welcome to email me or contact NPEU and express your interests. I haven't talked about today very much about the role of gut-brain axis in preterm babies. Clearly, this is a very kind of hot topic. Anything that's going to improve neck is going to improve brain outcome. HMOs do appear to be very promising intervention to reduce neck. We've got some data out, Massey et al. In gut this year, looking at the role of one of these HMOs, DSLNT. I haven't really got time to talk about it today. Let's not forget about the late and moderately preterm babies, those between 32 and 36 weeks. There are an awful lot of these babies, and as neonatologists, we tend to ignore them. But remember that the brain of a 34-week infant is only about 65% of that of a term infant. So how do we assess nutritional status? This is a really kind of quite a complex thing. I'm telling you that you need to think about all of these different aspects, nutrients and functional components and all the behavioral aspects. How do we determine whether our nutrition is good, and how do we measure nutritional status? Well, again, this would be a talk in itself, but if you want to have a simple way of approaching it, I would suggest using this ABCD approach, thinking about anthropometry, bloods and body composition, clinical aspects, then measuring the actual dietary intakes of the macronutrients, and then E, which is environment and evaluation, particularly thinking about, are we supporting the mothers to breastfeed? Is the baby ready to move to bolus feeds or ready to start breastfeeding? And this involves the whole team. And I've got a little schematic that prints out onto a couple of sides of A4. Again, if you want a copy of this, you're very welcome to email me, and I can send you a copy to use in your unit. But this is designed to be a simple means of assessing nutritional status that can be conducted by any of your bedside nurses or your trainees, or even any of you who are feeling a little bit uncertain about how to assess nutrition. And this only requires a calculator. It doesn't require any other books. This will only take you about 4 or 5 minutes. And it's really there as a prompt for you to decide, are you actually providing this baby with good holistic nutrition or not? It's not going to tell you exactly what to do. But say if you want to copy that, then ping me an email. So better nutrition, key to improve survival, but as survival increases, we're faced with more and more complex babies, and we need to be aware of their needs. Going to finish up now. What's the summary? So summary of nutrition interventions. Well, as I say, the strongest evidence relates to using mother's own milk. Very strong evidence for that. Pretty good evidence that meeting protein and energy needs are going to be important. But remember, there is going to be an upper limit of benefit. Just because 4 grams of protein might be better than 3, it doesn't mean you should aim even higher. So there's going to be a sweet spot, which is probably around about 4 grams. No evidence for some of these amino acids. Good theoretical evidence, obviously, for nutrients, metals such as iron and zinc. And when we think about the fatty acids, they are going to be key, but we need to get the quantities and proportions right. So if you want better brain outcomes, maximize mother's own milk and meet macronutrient needs. So why is breast milk so amazing? Well, when I started all the research, this is what I thought. I thought you just tip milk into the top end of this great big long tube called the gut. You sucked out the food and the fluid and the nutrients, and you produce waste product at the other end. Honestly, that's what I thought about nutrition 20 years ago. And we've now started to realize that, of course, it's much more complex than that. And as a research group, we're very interested in lots of these metabolites, microbes, and immune aspects. So we need to emphasize that nutrition is more than nutrients. And breast milk is more than simply food. There are lots of functional aspects here that we need to better understand. And here's my final diagram. On the left here appearing are all the 75 components that appear in formula milk. But now appearing in color are all the things that you get in breast milk that you don't really get in formula milk at all. So a whole range of individual amino acids, lots of different free fatty acids, lots of different lipid compounds, finger lipids, a whole range of cytokines, leukotrienes, prostacyclins, lots of nucleotides, so many we don't really know what many of these do. Growth factors, IGF-2, IGF-1, cholecystokinin, beta endorphins, your thyroid hormones, cortisol, prolactin, oxytocin, insulin in breast milk, what's that doing there? 200 different HMOs, a whole range of interleukins. So I think you get the idea that breast milk is really key to improving brain outcomes in preterm babies. And with that, I'm going to finish. As I say, we've got a presence on Twitter if you want to follow us. You can go to our website, neonatalresearch.net, and look at these slides again, or you can email me. And I'm going to finish there and say thank you very much.
Thank you. Thank you, Nick. That was amazing. Thank you very much. Very extremely informative. And we've already got a host of questions for you, I'm glad to say. So I'm going to kick off with, I think it's Dr. Jagged, I think. I hope I've said the name correctly. So asking, are sort of preterm outcomes related or associated with the etiology or the cause of preterm birth? And my take on that is, we know that for preterms it can be quite challenging to get them to grow. We know that 50 to 60% of our extremely low birth weight infants go home, still, weighing not that much. And so is it possible actually to do a good job on these babies? And is it just, it's a lost cause because they've been born preterm already?
Yeah. Absolutely. All of us could do better, including me. I could go to my neonatal unit now. I could get up out of here and drive to the neonatal unit, and I could make a difference to the nutrition of some of those babies if I really put the effort in. I think that, suggesting that this is all challenging and it's a bit sort of fatalistic and you can't do anything about it is the wrong approach. All of us can make a difference. The reason we don't make a difference is there are lots of other pressing priorities. You're busy sort of tubing the babies and people are focusing on lots of other things. But I think, that's part of the point I'm trying to make here, is that if people simply focus on the blood gases and the ventilator and the echo machines, all these kind of things, you're missing out on a really key intervention here. I was never taught nutrition at medical school. I don't know about the rest of you. It just didn't feature in my medical school training. It didn't feature in any of my SHO, my registrar training. I came to do my research thesis without even really knowing what protein was. I kind of had an idea that it was amino acids, but I literally knew nothing. And I was a couple of years off being a consultant. I don't know whether any of my consultant colleagues are on the line, but I mean, I know lots of consultants for the places. I think most consultants would admit you just don't know enough about nutrition. Most people really struggle to understand how much protein, energy, what's in a bag of PN. So undoubtedly, all of you on this call can make a difference to that to baby's nutrition on your unit today. And if you did that routinely and you focus much more on it, your outcomes would be better. I'm not pretending that you can resolve all of these problems. But yes, I think very much the worst outcomes we see in these babies can be improved by better attention to nutrition, no question.
Thank you. Yes, I completely second that. And I think sometimes in neonatology, the sexy subjects are the cardiovascular system and the respiratory system. And I'm sure my colleagues online will be laughing at me now because that's what I keep on saying.
I can't believe I see these people wheeling out these bloody echo machines.
All the machines.
To measure this duck with all these pretty red and blue colors. It's a very macho specialty. And you know, nothing looks sexier and impressive than an echo machine because nobody quite knows what that whooshy sound makes and those red and blue colors.
Yes.
To me, like, show me a single study where someone's got an echo machine out and improved brain outcome. They don't exist. Never seen one. Yeah, there's a lot of arguing in the background now, but honestly, people go to neonatology because they think neonatology and improving outcomes is all about, you know, using fancy technology. You spend all this time fiddling on with assist control, pressure support, you know, all this stuff. It makes no difference to the baby's brain. That baby's brain will not grow if you don't provide it with the right amount of nutrients. I know it's not very sexy, but that's the reality.
Absolutely. I think the cornerstone is nutrition, because if you don't get good nutrition, no matter how good or sexy your impressive cardiovascular respiratory knowledge is, if the baby isn't going to grow, then it's not going to be reflected. But I guess we're preaching to the converted, all of those people
you can't defeat the laws of physics. I mean, energy can't be created out of nothing. I mean, a brain can't grow out of nothing. I mean, the brain will not grow if you don't provide it with what it needs. I mean, it's not rocket science, unfortunately. Sadly, because it isn't rocket science, then most people tend to ignore it. But anyhow, that's
I quite like the idea of Nick's idea about the nutrition monitor. You know, I think we really should have a nutrition monitor next to bedside. You know.
Yeah, I think that's part of the problem is that we don't get good feedback, do we? I mean, the only feedback that you get from the nutrition system is growth generally. I mean, most of your blood work is irrelevant. And so you do get a growth chart, but you're also used to looking at babies who don't grow, that even when I show you a growth chart with the baby not growing, you still go back and get the echo machine out.
Okay, so there's a lot of sort of quite detailed questions, but there's one question that I wanted to ask actually regarding breast milk fortifiers. And there's obviously cow's milk fortifier, and you know, when do you start it? Do you wait for full feeds and then start it, or do you feed it, or do you start before reaching full feeds? And I know also, I think you have used or have been involved in the human milk-based fortifier, and your thoughts about sort of pros and cons of this, please.
Okay, we start breast milk fortifier once the babies have got to full feeds and maintain that for 48 hours. The Americans will study it early. There are no good trials telling you what's the best approach. My own personal feeling is that when you start adding breast milk fortifier, you do disturb the microbiome, you do have some other interactions, you create a few soaks in the gut. Maybe my own feeling is I'm better to get that gut as healthy as I can by giving it as much breast milk and getting that baby up to full feeds before I start fortifier. But honestly, there's no data. I think you could start fortifier early if you wanted to. The problem isn't starting it at full feeds. The problem is people waiting 2 or 3 weeks for the baby to stop growing before they start it. I think that's the biggest risk. Human milk fortifiers, it's a very interesting topic. I mean, I think we'd all love to think that human milk fortifiers was going to stop babies getting neck and improve brain outcomes. We have done some a decent sized research study. I haven't published it yet, so we can't share the data, but I think human milk products might have a very specific role in some babies. However, the studies that are currently done are simply not big enough to justify the expense. I think that would be my reading of the data at present.
Okay, lots of questions about micronutrients, specifically iron, when to start, and then do you monitor with ferritin, and also specifically for surgical babies and zinc. There was a bit of a debate going on about do you give more than the standard requirement, particularly for if there's a stoma.
Yeah. So again, the good studies haven't been done. Iron, I think it's fairly clear. You need iron for your brain to grow, and I think people forget that. I think when people are giving iron, they're thinking that it's stopping the baby getting anemic, but you need iron for your brain. Clearly, if you've got a 24, 25 weeker who you've transfused a couple of times in the first 2 weeks, they've probably got a decent amount of iron, and you can wait another 2 weeks. But if you're a 28, 29 weeker who never got a transfusion, and you're starting to feed and grow, you're going to need iron from about 2 or 3 weeks of age. So people need to think about it. We don't routinely measure ferritin. As I say, our biggest crime is to forget to start it properly, not because we don't want to give it, but because people forget about it, and that's the role of dietitians. So I think everybody's agreed, you need iron for your brain to grow. Don't give it if you've had loads of transfusions. If you're really not quite sure whether the baby's had too much blood, you can check a ferritin, but I maybe check 1 or 2 ferritins a year. I don't think it's something that you need to do routinely. Zinc, of course, zinc is involved in every single body process. Any enzyme needs zinc in it. So zinc's critical, but the evidence that currently that babies are zinc deficient just isn't that strong. I mean, I certainly think about it in babies with poor wound healing. I agree in babies with stomas, short gut, you probably need to think about it. It's relatively easy to measure a zinc level if you're not certain. Zinc is very safe and benign to give. You're not going to kill a baby by giving it a little bit too much zinc. It's not pro-oxidant like iron is, so it's a relatively safe thing to do. Just be careful that your micronutrient levels, if you measure them in a sick baby with inflammation, are going to be all over the shop. You cannot interpret serum levels in a sick patient. It's just impossible. But in a stable patient with a stoma who maybe has evidence of poor wound healing, absolutely measure a zinc level and give the baby some extra zinc.
Thank you. Some other questions then regarding lipid preparations, and as always, the devil is in the detail for lipid preparations. And is there a particular one that you would recommend? And do you ever use sort of would you ever, obviously I'm working in a surgical unit, and we have sometimes used a Megaven, which is 100% fish oil lipid. Is that something that you have experience of?
Yep. I think we all need to be a little bit careful with lipid. I think the temptation is to imagine that the box standard intralipid soybean is not good enough. Actually, the meta-analysis, the data suggests that if you're just wanting to give the baby some essential fatty acid and get regular babies to grow, it probably doesn't matter whether you're using intralipid SMOF or any other solution the first 1 or 2 weeks. So regularly for most preterm babies, you can probably pretty much use what you have. Clearly, we have a concern that soybean does not contain enough N3, and so a lot of people have been using, as you say, fish oil-based, olive oil-based products over the last sort of 10 years or so, with the kind of assumption, well, that has to be better for your brain. That there is no data to show that using these lipids improves brain outcome in the babies. There, of course, is evidence in slightly older infants that using SMOF might result in better liver outcomes. I would be cautious about using a Megaven. I think 100% fish oil has a very high EPA content, and we're just starting to get a little bit more concerned about that. And I think in the past we used to see, high icosapentaenoic acid intakes and think, well, it's probably benign. I think we're less sure now that it's a safe thing to do. So I wouldn't use pure fish oil for very long in babies. So we tend to use SMOF at the minute, which has got a bit of everything. But we need more studies. But as I say, don't be fooled into thinking that because something's more expensive and more complex that it's necessarily better. I don't think we know that yet.
Yeah, yeah. Can I ask a question, Nick, then, regarding protein and energy requirements? And in the latest sort of ESPGHAN recommendations, maybe it's more obvious for the term babies, but both sort of protein and energy recommendations have been lowered. And you know, in your talk, you were emphasizing the importance of micronutrients and getting that right. And so, is it balance? Is you know, can you get to, you know, is there an anxiety about too much protein, about too much energy, and you know, where is that balance?
Yeah, I could probably talk all day about this. It's a very complex topic still. I think what people need to appreciate is that parenteral and enteral are not equivalent. We can't just have a single amount of protein that you need and say it doesn't matter what you do, just give that amount. Humans have a requirement for amino acids. We don't actually have a requirement for protein. We have a requirement for amino acids, and clever old evolution designed the placenta to transport the right amount of amino acids. When you start giving high protein intakes like these babies need, if you're giving that from a nitrogen source that maybe isn't completely optimal, you may end up doing more harm than good. So for example, parental nutrition solutions are not really designed for 24, 25 week babies. I mean, they're a compromise. And so when you shovel in these parental amino acids, if you shovel them really at high amounts, you will get a few abnormal peaks. And I think we just started getting a bit concerned that people imagined, well, I just have to give 4 grams of protein a day. It doesn't matter what I'm giving it. And if you give 4 grams of some PN solutions, I think you might find some slightly abnormal amino acid peaks. I'm not saying it's definitely dangerous. I just think we need to be a little bit cautious, and that's why we've knocked it down a little bit in the recent ones. I think in regular babies, preterm babies, if you're getting them up to somewhere between 3 and 3.5 within the first 3 or 4 days, that's probably enough. You might want to finesse it for the very complex, small, long-term babies, but I think regularly you don't need to aim at 4. And then when it comes to protein in entry-fed babies, remember really what babies want is the amino acid composition that would come with 4 grams of breast milk. Now, when you give breast milk plus fortifier, you're giving about 2 grams of breast milk protein with a beautiful composition of amino acids. And then the other 2 grams you're making up with a fortifier, which is actually hydrolyzed whey. And hydrolyzed whey has a different amino acid composition to preterm breast milk, much higher in threonine and things like this. So again, I think we're safe giving 4 grams to most babies, but I don't think you can necessarily infer that giving 4.5 or 5 grams routinely to babies is going to be a good thing. It might be fine, but there's going to be a sweet spot. And so, you know, if you want to pick a number, I would say aim at 4. In a bigger baby from 30 weeks onwards, somewhere around about 3.5 is going to be all right. In a baby at 24 weeks, you might want to go aim a little bit higher than 4, but I think people need to be cautious about pushing them right to the top end in all of your babies.
Then can I ask, someone else has also mentioned this as well. So if you're on the unit and there's a new 24-weeker that's been born, and there's no EBM available, and time is marching on, and it's day 2 and it's day 3 and it's day 4, and you're still waiting for a colostrum and EBM. When do you bail out and say, I'm going to give something, and what is that something? Is it formula? Is it donor EBM? I mean, not all of us have availability of donor EBM. Certainly in Qatar, we don't. So I'm just interested in what's your sort of personal practice in that scenario.
I mean, we can get 95% of our mothers to provide some breast milk. It's very unusual for us now to have a mum who doesn't provide any. I'm aware that you're in a different kind of cultural setting, but if you really can't be getting 90 to 95% of your preterm mothers providing at least some colostrum, there's a problem in your system that needs attention and fixing. That's what I would say. Like, it just has to be possible with compassionate, empathic, thoughtful support. Most mothers get the importance of providing at least some colostrum and breast milk in the first few days. So if regularly you're not getting anything from lots of mums until day 3 or 4, then that bit of the system needs to be looked at. if I did have a mum, what I tend to say is I don't worry about the first 48 hours. I don't want mum to feel pressurized that if somehow she doesn't produce EBM that I'm going to start giving the baby milk. But I would personally be uncomfortable waiting beyond 72 hours of age if the baby's otherwise well, because then you're accepting all the risks of having PN and a slightly kind of rubbishy, you know, amino acid solution. So for me, if there's no breast milk between 48 and 72 hours of age, I would ask the nurses to speak to mum. I would say, look, we think we're better off starting a little bit of something else. It doesn't mean you still need to keep on expressing and trying and all those kind of things, but that would be the time that I would do it. Of course, we would use donor milk. I'm not absolutely convinced it reduces NEC, but it's what mothers tend to want. And, you know, if mothers want it, and I think it's not going to be harmful, I think I have a duty to listen to what the mothers are telling me. If you haven't got donor milk, it actually doesn't really matter what you use, whether you use a preterm milk or a term milk or hydrolyzed milk. There's absolutely no evidence that any of those formulas pose a greater or lesser risk for NEC. So if you can't get donor milk, I would just use whatever you have on the unit. I don't think it honestly matters.
Okay, thank you. I also want to ask you regarding growth. So what would you, there's a question here regarding how would you recommend that we monitor growth? Should it be weight gain or growth velocity? And also asking regarding what sort of a BUN level that you would look to sort of give you some sort of reassurance that the protein intake was adequate.
It depends on how much time and effort and how many people you have around you to help. I mean, if it's just you standing down the ward round and you haven't got anybody else doing this work for you, then I tend to remember that bigger babies should grow about 30 grams a day and smaller babies should grow about 20 grams a day. That's a very crude, rough number that works. Clearly, the more accurate way is looking at grams per kilo a day, but you've got to recognize that if you use grams per kilo a day, the rate that is appropriate for a 24-weeker is about 21 grams per kilo a day. By the time you get to 32 weeks, it's probably going to be around about 15. And if people go to my website and look at my slides, I've got those in tables and some of my other talks. However, the easiest way of monitoring growth is just to stick it on a growth chart. I mean, most of us now have electronic medical software, and you just need to plot the points on a growth chart, stand back and look at what's happened over the last week. I don't think you really need to spend a lot of time working out grams per kilo a day. If the points on a growth chart look like they're following the centile, that's probably enough. So I would accept now that it's normal for babies to move down about half a standard deviation, somewhere around about 1 centile position on a standard growth chart. That's because there's an inevitable contraction of your extracellular fluid in the first few days, so you're going to have to lose a little bit of weight. But after that, we should be aiming to keep the baby on that centile line. I don't think there's a huge advantage to making babies catch up routinely. I think they can just stay on a slightly lower line, but they should then stay on that line until they go home. If they're drifting off the line, then you need to think about it. And as I say, that's where, if you use that little ABCD approach, it gives you a sort of schema to think about it. And then BUN, yeah, there are people who love BUNs. You know, it's good fun. It's another number. It's a bit easier to get your head around the number than some of this kind of other stuff. It's true. If your BUN is less than 3 millimoles a litre, you're probably not quite getting 3 grams of protein in. So if you have a baby growing slowly with a BUN less than 3, probably the baby needs a bit more protein, but he might just need regular fortifier. If your BUN is over about 6, 7, 8, then you might be pushing the protein a bit high. So there is a nice relationship between BUN and protein intake, but around that, you know, correlation, there's a lot of variability. So you might have a baby with a BUN of 4, and that baby might either be getting 2 grams of protein or 4 grams of protein. It's just not precise enough for you to know. But I mean, if you like looking at numbers and you want to impress people, then ask for a BUN and scratch your head a bit and look clever. But I don't think we really know how to use it.
Okay, and one last question, Nick, because I'm aware of the time. Can I ask you about the Sift study? So looking at differences between increasing at 18 mils per kilo per day versus 30 mils per kilo per day. There's lots of it, very large study, very well done, lots of interest in it. But in the end, there was no difference found between, at the long-term outcome and neurodevelopmental delay. So that was interesting and possibly disappointing. So what's your personal practice? What do you take from that? And this is a common question every single day on ward rounds. So I think we'd be very interested to see what your point on this is.
Yeah, there were lots of lessons in the Sift study. One of them is that there's, that's a very expensive way of finding out no difference between the studies, between the two arms. Yeah, it was a pretty hard study, but amazing. And I think it shows that a neonatal community can get together. We recruited 2,800 babies ahead of schedule. So, the great thing about being a neonatologist is people want to work with each other. It's really enthusing that we can do these studies, and we need to do more of them. What the Sift study tells us is that in a typical neonatal unit, if you give the babies 2 or 3 days for the mum to start producing a bit of breast milk, you're around about day 3 or 4 before you really want to ask the question, how quickly do I go up? I don't think you need to start increasing milk feeds before day 3 or 4 in the vast majority of preterm babies. What it says is if the baby is otherwise okay, it doesn't matter whether you go up at 30 or 18. And I think that a pragmatic clinician would look at that and go, do you know what, in the bigger babies 28 weeks and over with plenty of EBM, let's go up at 30. In the 24-weeker with absent reversed end diastolic flow, who's had a bit of sepsis and everything else like that, I might go up at 18. So I think it gives us the flexibility to realize when we write these guidelines for a neonatal unit, a 24-weeker and a 32-weeker are two completely different beings, and you're not going to have a single rate that works for all of them. I think it also tells us that if you want to go a bit more quickly, you don't have to worry that going a bit more quickly increases the risk of neck. It doesn't increase the risk of neck. So you can go up more quickly in a baby who's tolerating it without worrying about increasing the risk of neck. It also says that if you run a good neonatal unit, 5 extra days of PN, 5 extra days probably of a central line, does not increase your risk of sepsis. So if on that specific day, the nurse is saying to you, look, I'm just not happy about him, his tummy is a bit distended, there's something just not right, you don't have to keep on forcing that baby to keep more milk. You can just stick where you are for 24 hours and then come back and review the baby tomorrow. So I think it tells us, do you know what, TPN long lines in the right hands are not poisonous or toxic. It's okay to have another couple of days in the risky babies. You're not going to increase your risk of sepsis.
Okay, thank you very much, Nick. That was excellent, so interesting. And I'm sorry we weren't able to get to everybody's questions. I hope that we managed to answer most of them. So it was an outstanding lecture. Thank you very much. I'm going to hand over back to Kira now. Thank you.
Perfect. Wow, thank you, Nick, and thank you, Sybil, for moderating. It was a really excellent session, and I can see the enthusiasm from the audience. Actually, so many questions are coming. Looks like we need another session on this topic.
Well, I'll come back if you invite me back. Then I'll come and do another one.
Drive you back, for sure. So many questions. I have taken screenshots.
Yes, please. More nutrition. More nutrition.
Yes.
It's the only thing that matters. I mean, none of this echo stuff makes any difference. I don't know why people get so excited about it.
Okay, let's not get into that. But anyway, definitely, lots of questions. I'll see if I can collect this question and try to answer to individuals. But I think considering the time, we'll call it end to the meeting. Thank you everyone who has attended, and thank you very much again, Nick, for sparing the time. And it was a great, very comprehensive talk on nutrition. I think it was amazing. No words for it. So anyone else has any comments or final words? There were a few people also have joined with some nutrition interest. I can't.
I put, just to remind people, I've got this website, neonatalresearch.net, and if you go there, I've got a tab for talks. I think it's called Talks and Presentations, and I've got a whole load of PowerPoint slides and videos, and there will be a video on this type of lecture, not this exact one, but there'll be something like that. So people can go back and look at these slides again, and you can use those slides, you can give them to your fellows, your trainees. It's all completely free. From the love of Newcastle to the world of neonatology, it's all there for you if you want it.
Yeah, I've recorded the talk and I'll share with you, Nick, as well, so that you can put it on your website, and I'll share it individually in the social media groups as well if you are open.
Okay.
Perfect. So once again, thank you everyone, and thank you Nick, thank you Sybil, and all of those who have attended. Okay, see you next time,
Okay.
next
All
month.
right. See you next time.
Thank you.
Bye.
Bye. Bye. See you.
Thanks, Nick. Bye-bye.
All right. See you.
The intellectual